Hello everyone. A 2 years old male child has been admitted to your hospital with multiple episodes of pneumonia since last 2 years. When you examine the child, you find that child have multiple bruises all over the body and the skin of the baby is light in color and there is silvery lust hair present in the baby. The child also developed ataxia at 9 months of age. When you do the bone marrow study, you find that neutrophil having large inclusion body. So what may be your diagnosis? The options are related to Chediak Higashi syndrome, HLH or Gracilli syndrome. In this question, we found that the child is suffering from multiple episodes of pneumonia for last 2 years. That means child having suffering from primary immunodeficiency. And also we found that the child is having multiple bruises all over the body. So this primary immunodeficiency is associated with platelet problems or bleeding problems. And the child having lastly silvery hair and light skin. That means ocular problem. And the child is also having ocular and cutaneous problem. And the child also having develop ataxia, that means neuropathy. When we do the bone marrow examination, we found that neutrophil having large inclusion body. What do you mean by inclusion body? Inclusion body means the lysosome get attached to each other and produce a big lysosome, but it cannot kill the bacteria which is enter into the body as phagosomes. So phagosomes are not attached with the lysosome. Instead of this, the lysosome get adhere with each other and forming a big molecule that is called large inclusion body. So what is your diagnosis? This is a diagnosis of Chediak Higashi syndrome. Today we will discuss about the Chediak Higashi syndrome. What is this Chediak Higashi syndrome? So it is an autosomal recessive disorder, and the Chediak Higashi syndrome is occurred due to List gene mutation, and this gene is present in the chromosome number 1q. In this Chediak Higashi syndrome, there is oculocutaneous problem. That means the skin problem is there, and the child having suffering from neuropathy. That means nerve problem is there. The child is suffering from platelet problems, and the child is suffering from neutrophil adhesion disorder, and neutrophil cannot kill the bacteria. How this Lees gene causing this, I will discuss in later on. So the clinical features is child having light skin, that means albinism. Child is suffering from albinism, silvery hair, and child is having photophobia due to albinism. And the child is suffering from neuropathy. Mostly the neuropathy is sensory and motor neuropathy is there, and child is develop ataxia after 2 to 3 years of age. And the child having bleeding problem with normal platelet count. So platelet count is normal, but there is a bleeding problem. That means the platelet aggregation problem is there. So how this List gene causing this type of disease? Now, if some bacteria enter to your body, then this bacteria firstly taken by the dendritic cell or NK cell or some macrophages. This bacteria then engulfed by the body by endosome and causing, and this is produce a phagosome, and this phagosome now attached with the lysosome, and the lysosome produces some massive cytokines, that means histone or myeloperoxidase or some other cytokines, and kill the bacteria. This is how the body kills the bacteria. In List gene protein defect, the List protein is not due to List gene mutation, the List protein is not formed. So what happened? The List protein is present in the lysosomal body. Suppose this is a cell and this is lysosome. This lysosome body having some List protein. This List protein, what it will do? It will attach with the phagosome. When the bacteria enter into the body by phagosome, this phagosome and this lysosome have to be attached each other to kill the bacteria. So this phagosome it attached with the lysosome by the least protein. So if the least protein defect is there, the phagosome is not attached with the lysosome, and due to the bacteria enter into the body, there is a multiple cytokines release, and these cytokines make the lysosome to adhere each other, but it cannot adhere with the phagosomes. So that's why there is inclusion body we found. That means big lysosome we found seen in the neutrophil, but this neutrophil, this lysosome cannot attach with the phagosome, and that's why the bacteria will not be killed by the body. So there is defect in the Lees protein, and there is a recurrent infection caused by the staphylococcus infection, streptococcus infection, or other infection, because this Lees protein defect, the lysosome cannot kill the bacteria, and this Lees protein also causing problem in the defect of the platelet ATP ADP formation. Due to ADP formation, the platelet is not adhered each other. That means in the blood vessels, if there is any minor breaches present, then platelet get adhere with this endothelium tissue, and there is a formation of a platelet shield, and that's why the bleeding will not happen in this in normal patient. But in Lees gene protein defect, the platelet is not adhere with the endothelial tissue as because of the ADP aggregation defect. So platelet will not aggregate each other. So there is a no platelet shield is formed, and the blood is trickled out, and this causing bleeding problem, that causing increased bruisability. In the nerve, the dendritic cell is mainly of the body is adhere with the axons of the body and the dendrons of the body. Due to the lesion defect, the dendrocyte are not covered the axons, and that's why the axon get exposed with the normal virus or bacteria, and this causing neuropathies. And due to this, the peripheral neuropathy, that means sensory neuropathy, is there because the dendritic cell is killed by the normal common cells, and that's why there is neuropathy, and patient causing weakness, ataxia, or patient may develop also seizure. This gene also causing melanocyte storage defect, because the melanocyte is one type of macrophage. As I say, there is a problem in the neutrophil and also the macrophage gene. Melanin present in the melanocyte will not get attached to the keratinocyte present in the upper layer of the skin, and there is a due to defect of the keratinocyte melanin. Due to the defect of melanin in the keratinocyte, the child is developed albinism. So these are the typical features of Chediak Higashi syndrome. So in a nutshell, due to the Lees gene defect, there is formation of inclusion body, and the lysosome is not attached with the phagosome, and due to this defect also the melanocyte is not enter into the keratinocyte and causing ocular or facial albinism. Now the diagnostic investigation is genetic study. It is a gold standard study, and we can also do bone marrow study to see the inclusion body. And the treatment of choice is hematopoietic stem cell transplantation. This is the all India MCQ, and we also can use high dose of ascorbic acid. So this is also an MCQ. So if you found a patient having multiple episode of pneumonia and the child is albinism, and the child having some bleeding problem and also neurological problem, then think about Chediak Higashi syndrome. In Grassley syndrome, in our option 4, this also one type of HLH. In Grassley syndrome, these all features are seen, and along with this, there is specific point in Grassley syndrome is there is hepatosplenomegaly. So if you found hepatosplenomegaly along with this all features, then think about Grassley syndrome. In lab 2 defect, we can found this type of problems, but there is no ataxia present in the child. The child do not have any lustrous silver hair or light skin. So in lab 2 defect, there is a typical Bombay blood group is present, and there is a delayed separation of cord.